this video we will discuss about a new paper where they have developed the ex vivo extracellular matrix, which is composed of these peptide amplifiers and ECM, and it is highly tunable and it can give results similar to the in vivo observations. So in this video we will discuss about the characterizations and the design of this PA-ECM. PA is peptide amplifiers and ECM is extracellular matrix. So this is the components of their PA-ECM, which is co-cultured with this PDAC, that is pancreatic cancer cells. It composed of these pancreatic cancer cells, these pancreatic stem cells, and the macrophages that can be seen here. PA-ECM co-assembly. And when they check their electron microscopic images, they can see that it can form the fibers. When they use this collagen and fibronectin, they can see that thin fibers are formed, and when they use only the collagen, they can see that thick fibers are formed. And these things can be used to tune their cellular matrix. And when they use only this PA and ECM, they can see the fibers are still there without any collagens and fibronectin, and these fibers behave differently compared to the case where collagens and this collagen and fibronectin both are introduced. So this indicates that their newly designed extracellular matrix is highly sensitive to the different kind of substrate that is used with it. And what are the current PDAC in vitro models that are available? First one is this 2D monolayer models where pancreatic cancer cells are just labeled on the polystyrene substrate, or we can see the cell culture model. Another one is the spare model where no substrates are used but spares are formed. The third one is this organoid model where matrigel substrate is used of different stiffness. Matrigel substrate is used and the organoids are grown inside this matrigel substrate. So these are the current PDAC in vitro models used, and they have designed a 3D models in vitro models that can be used to, and those results are in line with the in vivo observations in the patients. And the further characterizations that they have done is that the size of this PA, that is peptide amplifiers, is similar to the is around this in terms of the length. It is around 250 to 450 nanometers, and in terms of the diameter it is around this 20 nanometers, and this size also in line with the regular substrate that are found in the pancreatic cancers extracellular matrix or tumor microenvironments. And when in terms of their beta sheets characterizations, they can using the dichromes, which is used to characterize the secondary structures of the protein. They find out this that beta sheets are the peptide amphiphyllins has the beta sheets where the signals, we can see the signals between 200 to 220, and these are the signals where which are usually observed when there is a beta sheets in the samples. And in terms of the stiffness also, we can see that stiffness of this PA-ECM is in line with the in vivo stiffness that are found, that is around 1000 pascals, whereas matrigel stiffness is very low, is around 90 pascals. So indicating that this PA-ECM stiffness is in line with the stiffness that is observed in the pancreatic cancer patients, and this stiffness can easily be modulate using some gelation media to increase or decrease, using some gelation media to increase the stiffness, as in some cases stiffness of the patients are found to be higher at around 10,000 pascals. So these things can be achieved using the gelation agent, and the process that they have used to test their 3D developed PA-ECM is that first they developed this PA-ECM co-assembly and then co-culture this PDAC in the PA-ECM. Thereafter they did this transcriptome analysis, and this thereafter from the transcriptome analysis they will check this cancer stem cells analysis and also this matrisome analysis. And then once they have the information about this cancer stem cell analysis, then they can tune this extracellular matrix in a way that these drugs can be used to target these cancer stem cells. And once they have the informations about this tunability, then they can use those informations in the on the patients. And one more advantage of this approach is that this approach can be used or customized based on patient to patients, so that as each patient has different degree of pancreatic cancers and has different kind of stiffness around the pancreatic cancer cells based on the mutations that has occurred in the patients. So these things can be customized or tunable to the individual patients. Therefore, it might give much better performance and provide a 3D in vitro tumor microenvironments model to study the pancreatic cancer cells behavior. Yeah, I hope you understood the basic characterizations and design of PA-ECM that is mentioned in this figure. If you need to understand more about their PA-ECM designs, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.